Yes, hello, Dr. Valentina Viduta, and I am founder of Long COVID Foundation. Today I'm a panelist, and I will help you to learn on gut-brain axis and learn more about the bacteria. So looking forward to it.
Thank you. And Mike?
Hi there, I'm Mike VanElzakker. I'm a neuroscientist at Massachusetts General Hospital and Harvard Medical School. I'm going to talk a little bit about neuroimmune communication today.
Okay, thank you. Joshim?
Yeah, very big welcome from my side. I'm excited for the second conference we are holding today, and I'm a panelist today. I'm head of and will ask a lot of questions today and not speak much.
Hi, I'm Leo Galland. I'm a physician and an independent researcher in the US. I'll be speaking today about the gut-brain connection in COVID-19 with an emphasis on the gut microbiome.
Excellent, thank you.
Hi, I'm Dr. Shankara Chetty from South Africa. I'm a natural science biologist and a frontline worker. I've been looking at COVID from a clinical perspective from the start of this pandemic and hoping that we can get more insight into long COVID.
Excellent, thank you. And Stephanie?
My name is Stephanie Seneff. I'm a senior research scientist at MIT. I'm the author of this book, Toxic Legacy, How the Weed Killer Glyphosate is Destroying Our Health and the Environment. The book talks a lot about the gut microbiome and interaction between the gut-brain axis. I'm a panelist, so I'll just be asking some questions today, but I'm looking forward to hearing the presentations.
Excellent, thank you. And we have Carlo.
Hi, I'm Carlo Brogna. I am a doctor in the research of Craniomed Group Italy, and today I will speak about the Toxin and the SARS-CoV-2 and long COVID condition.
I'm Abdul Manan Beg. I'm the lead researcher of infectious disease group at Aga Khan University. Today I'm invited as a panelist in the second coalition conference. Thank you very much.
Excellent, thank you all very much. Yes, so appreciate that from everyone. And so moving right along to what is quite a fascinating area of research with regards to long COVID, we have got Mike VanElzakker. Have I got that right, Mike? You're muted at the moment.
Yes, yeah.
Excellent. And so I'll not hold you back, Mike. I'll let you go straight away into your presentation, at which point I will then bring in your panelists to have a discussion with you with some of the questions.
Okay, great. And
I'll bring up your screen for you. 1 second here. Is your screen still shared, Mike?
I am sharing. Can you see it?
Okay, let me just add it. There you go.
Is it sharing?
Yeah, it's sharing now. Got it.
Okay, great. So yeah, I appreciate the invite, everyone. So I'm going to talk about sickness and the brain, the neuroscience of how we feel infections and inflammation. It's sort of going to be a basic neuroscience, neuroimmunology talk. Sort of broad and mechanistic and anatomical, just explaining some of the reasons why people feel the subjective feelings that they feel when they're sick. And I don't have anything to declare, and my presence isn't an endorsement of anyone else's statements. I actually have COVID right now. I tested positive, I guess, 3 days ago, so I feel pretty sick. I'm going to try and get through this. I'm triple vaxxed. But I had a breakthrough infection like so many others are having. We all know about long COVID at this point. It's something that has really, been a topic in general media and around the world, our friends and loved ones. It's a serious issue. And it's something that we're going to have to really dig in and understand. So I thought that talking a little bit about the basic neuroscience of sickness and illness might be helpful here. This is a study from Hannah Davis and her collaborators. It's a group of essentially patient scientists and patient advocates, I suppose, where they followed people with long COVID early on and talked about the symptoms that people were experiencing 7 months after their onset. And the symptoms here are sort of centered around fatigue, brain fog, essentially the feelings of being, the very feeling of being sick, the sickness feelings that we all have, whether it's a flu or a strep throat, bacteria or virus, the feeling of being sick. Autonomic problems are really common. Some recent studies showing about 2/3 of people with, the long-term consequences of COVID showing autonomic problems and pain. So I'm going to stay focused on those categories of symptoms: sickness, autonomic problems, and pain. Sickness, autonomic problems, and pain. And we'll talk a little bit about those. Many of us have been working in the world of myalgic encephalomyelitis for many years. This is a phenomenon we've seen before, in which an acute infection, there isn't a full recovery, and people feel sick sometimes for many months or many years afterwards. Early on, we sort of knew that this was going to be, in a minority of people, but still a significant minority, and people that deserve healthcare. So if we look at the symptoms of myalgic encephalomyelitis, it's very similar to those symptoms that I just talked about in the long-term consequences of COVID. Now, with COVID, there's going to be some people who have lung damage, tissue damage. They were in the ICU and had a rough intubation. And those are particular types of insults, problems that aren't quite the same thing as ME/CFS, but there's going to be an overlap with ME/CFS. And again, if we look at the symptoms of ME/CFS, without going through them all, this is the international consensus criteria. It's striking that they're also focused on sickness, autonomic problems, and pain. Sickness, autonomic problems, and pain again. If we look back at when the term myalgic encephalomyelitis was first coined, in a 1956 Lancet article, in which the clinician at the time wrote into the Lancet saying, look, we've had these outbreaks. If you look here in the middle column here where it says virus, some of the times we knew what virus was causing the outbreaks, and some of the times we didn't. This is before PCR was invented. And yet it was causing this long-term problem. The term benign here just meant that it wasn't killing people like polio sometimes did. But it wasn't meant to say that it was minor, just that it wasn't killing people. So what you can see here basically is that several different kinds of virus, to the extent that they knew, were causing these same consequences, or apparently similar viruses are causing the same consequences. It's worth noting too that there's other the word post in big quotes, I think, post-Ebola syndrome here, for example, where people have an acute Ebola infection, they survive, and then they go on to have symptoms that include musculoskeletal pain, headache, in this case, ocular problems, but centered on those same symptoms. Post-Zika, a similar virus to SARS-CoV-2, again, causing somewhat similar symptoms. And here, the SARS-CoV-1 causing relatively similar symptoms, again, sort of focused on sickness, autonomic problems, and pain. People just describing these same symptoms. So we wrote about this a little bit in a review article early on, trying to make the world understand, for one thing, that this isn't quite as mysterious, I think, as we sometimes hear. We do have some understanding of what the mechanisms are. We do have some understanding from, for example, post-Ebola, quote unquote, that in that case, the Ebola virus was still around. When they finally tested in sanctuaries, they tested semen, and even in the eyes, there was live viable virus in those cases. And there's some accruing evidence that the same may be true in so-called post-COVID. So given that I named a bunch of different viruses, how can these disparate problems drive similar symptoms? How can we end up with the same sickness, autonomic problems, and pain from different causes? Well, I'd like to focus in on an area of the brain called the brainstem. And here's a scan of my brain at 7 Tesla. And I'm focusing in on the brainstem here. If we blow it up and Photoshop out the vasculature, it looks like this. And here's the anatomy. It's an area where cerebral spinal fluid flows through very thin aqueducts nearby. And I'm going to blow it up even further so that we can sort of focus on these anatomical nuclei within the dorsal brainstem of the brain, because I think it's important for the symptoms that we see and a mechanism for why we may see these symptoms. There are a bunch of structures and circuits that are in really close proximity, and they overlap and crosstalk. They communicate with one another, and they probably drive some of this, the subjective feelings that we feel when we're sick. So here, sort of centered, that I've highlighted in red now, is the nucleus of the solitary tract, NTS, nucleus tractus solitarius. And this is a place where the vagus nerve enters the brain from the body. Okay, so the vagus nerve is the 10th cranial nerve. I'll talk about that in a little bit. It shares the same Latin root as the word vagabond, right, meaning to wander, which means that it wanders all over the trunk, all over the torso. It innervates the lungs, the bronchia, the gut. If there's inflammation or infection happening in the lungs, if there's a dysbiosis in the gut, the reason that the brain knows about that is because the vagus nerve detects it. And this is where the vagus nerve enters the brain from the body. It's where a majority of these afferent or sensory vagus nerve cell bodies live. So nearby the nucleus of the solitary tract is an area called the area postrema. So this is a circumventricular organ without a complete blood-brain barrier. In other words, there are some areas of the brain where there's what we call a fenestration, or a window in the blood-brain barrier, which is sort of a barrier to prevent the blood from having easy access to the tissue. This is an area where there's windows in that so that the brain can sort of sample larger molecules. It's really important for vomiting and nausea. A lot of patients have ongoing problems with nausea with these complex chronic illnesses. These areas here are both body to brain and brain to body. So the two areas I talked about represented the body communicating to the brain, but now I'll talk about the brain communicating back to the body. So the dorsal motor vagus is the vagus nerve going in the other direction. So it's an autonomic nuclei. It's the site of parasympathetic cell bodies, or soma. And it's the main source of how the vagus nerve innervates the trunk organ. So the vagus nerve has control over, for example, heart rate. So if there's a problem with tachycardia, high, fast heart rate, even going from lying down to standing up, that may be an important part of why that's happening, because of this structure here. The dorsal reticular nucleus down here in red is important for inflammatory pain transmission. Now notice what I've talked about so far are sickness, autonomic problems, and pain, right? So the very feeling of being sick is the nucleus of the solitary tract, which I'll talk about in a little bit. The autonomic problems are represented at least partially by the dorsal motor vagus. For example, the tachycardia, fast heart rate. And here's a nucleus that's really important for the transmission of pain. So if there's a situation where we call hyperalgesia, in other words, where something that would normally only hurt a little bit ends up hurting a lot, we call that hyperalgesia. Or something that shouldn't hurt at all ends up hurting, that's called allodynia. And that may be important in this structure here. The ventrolateral medulla is involved in autonomic control of blood pressure and breathing. So again, autonomic problems, to the extent to which they exist in patients, may be sort of centered on these nuclei right around here. And the nucleus ambiguus right here is, again, an area that's important for controlling heart rate, which we know a lot of people with so-called long COVID or PASC have problems with tachycardia when they go from lying down to standing up, for example. Some people qualify for the POTS diagnosis, not all, but some people just have autonomic problems. So, I talked a little bit about the vagus nerve, and I'll talk a little bit more about that here. So again, it wanders all over the body. I've drawn a little diagram here from an older paper showing the different ganglia and paraganglia that are all over the body, really, all over the trunk. The vagus nerve innervates the bronchus, the lungs, very densely. So there's a lot of vagus nerve terminals at the lungs. So this is really important because inflammatory molecules like cytokines, you've all heard of cytokine storm, right? You know, the virus obviously triggers cytokines. Well, cytokines are relatively large protein molecules that don't easily cross the blood-brain barrier. So they don't just flow into the brain like a steroid hormone does. Well, how then does the body know that there's cytokines? Well, it knows because of the vagus nerve. There's chemoreceptors on the vagus nerve that detect the presence of those cytokines, and it sends a signal up to the brain that says, hey, guess what? You've got inflammation. You're sick. And that is the reason that we feel sick. It's the body's behavioral response to inflammation saying, you need to preserve your energy, go to bed. You shouldn't be spending your energy on jogging or other pursuits right now. You need to go to bed. So interestingly, for example, if you inject inflammation into a rat, the rat will not run on a wheel anymore. They won't eat as much, etc., unless you've cut the vagus nerve. And then the rat will act normally, even though you have injected inflammation, because the brain doesn't know that the body is sick. So one of the things that the vagus nerve does is to detect peripheral catecholamines. So, for example, epinephrine or adrenaline. You've heard of an adrenaline rush that's produced, you know, right on top of your kidneys, the adrenal on top of kidneys. Well, again, that doesn't actually flow into your brain very easily. It's the vagus nerve that detects those chemicals and signals to the brain. And it also detects local immune signaling molecules in the way that I've just described. So cytokines, inflammatory proteins are detected by the vagus nerve. Now, importantly here, because the vagus nerve wanders all over the trunk, the inflammation can be local. So you could have a gut dysbiosis, for example, inflammation in the gut. And it doesn't even have to be inflammation flowing through the entire circulatory system. It doesn't have to spill over into the blood, for example. It can be local. And because the vagus nerve has so many branches, it will detect that. And that will drive the feeling of being sick. So it surveys the gut microbiome. That's a major job. And it initiates the central illness response, which includes, as you might guess, the feeling of being sick, autonomic problems, and pain. So these ongoing symptoms that people may feel from different forms of illness. And we can measure that with PET scanning, brain scanning, which is one of the things that my lab is doing right now, is studying this in so-called long COVID, measuring inflammation in the brain. So when there's immune cells in the periphery, here I've drawn a monocyte and a macrophage, they don't easily cross the blood-brain barrier. So I want to talk a little bit about how the signaling happens. When these immune cells detect either pathogens, that's a virus, a bacteria, they activate. They sort of turn on, and then they produce cytokines. Now, one way that they could affect the brain is if they flowed right into the brain through the blood like a hormone does. Now, that doesn't really happen all that much. It happens a little, but not all that much. So another way that it happens is crossing the circumventricular organs. There's about a half a dozen of those circumventricular organs, including the area postrema, which I had mentioned before. So the cytokines, inflammatory molecules, can flow across those circumventricular organs and affect the brain and cause the feeling of being sick. Or interestingly, the immune cells themselves can be taken across the circumventricular organ, and they can enter the brain. This is actually a way that some pathogens have evolved to have a Trojan horse mechanism for infecting the brain. They infect the immune cells, and then the immune cells are brought into the brain. But another way that these cytokines can affect the brain is when they're detected by the vagus nerve. It sends, and because the vagus nerve is all over the place, it can be local, and it sends a signal up into the brain that triggers an inflammatory response on the brain side of the blood-brain barrier. So it allows local immune signaling to have a broad effect. It doesn't have to be infectious. It can also be injury. So, for example, if there's lung damage from the virus, even if theoretically the virus was fully cleared, you could have ongoing inflammation just from the tissue damage. And it triggers this mirror response of glial activation on the brain side of the blood-brain barrier. And this is something that they've measured in chronic fatigue syndrome in some studies, not others, and that we're studying in long COVID in my lab. I wanted to talk just for a minute about those glial cells. Glia means glue in Greek. It used to be thought that it was just holding neurons together. This is microglia of the brain. So when they detect pathogens or inflammation, they change their shape, they activate, and they produce, they actually change into this sort of different form, and they produce a bunch of things that excite neurons. So they produce cytokines, which I've represented with the little triangles, and they also produce things that excite neurons, which I've represented by these little lightning bolts. Things like glutamate, bradykinins, prostaglandins, ATP, nitric oxide, lots of different things that excite neurons, and a protein called the translocator protein, which we can measure with brain scanning. After that happens, the glia enter this state called priming. They're primed. What that means is that they're sort of hypervigilant, I suppose you could say. And if a second immune response comes and affects those glia, then the second response is even bigger. That's what I'm showing here. The second response is even bigger. So one of the things that I think can happen is that an initial inflammatory response from an initial infection, say the SARS-CoV-2 infection, can prime the glia, and then the glia becomes sensitive to things like gut dysbiosis. So someone that might have a little problem with gut, if they don't have this problem, maybe they won't even be affected. But if they have primed glia, then maybe they will be affected. And that can drive sort of brain fog symptoms. You know, I talked a little bit about that area in the brainstem, the nucleus of the solitary tract. If there's glia that are excited there and they're turning on neurons nearby, well, look at all the nuclei that I talked about that were nearby that control sickness, autonomic problems, and pain. So it's almost like having a little amplifier on these nuclei. Now, that's not to say that we should immunosuppress these patients. I'm not sure that that's the right call. What it means is if there's something driving inflammation, whether it's a gut dysbiosis, whether it's other infectious, whether it's injury, whether there's viral reservoirs, that those things need to be addressed because there's a sensitized system in people that make them extra sensitive to insults, including things like toxins and infections. I've talked a little bit about this in a previous paper that you may be interested in learning more about neuroinflammation. You can look at it here. And I'd just like to thank my collaborators that helped me with all the brain scan projects that we have going, including in long COVID. Thank you very much.
Excellent. Thank you very much, Mike. I really appreciate that. And again, I'm seeing so many parallels with all of the presentations and seeing how they all interact and come together. Again, I'll have to save my questions for later, but I'll bring in here your panel. We've got Shankar and we've got Manan here, and I'll leave us here for about 15 minutes or so, and they can ask you some questions, and then I'll come in and we'll move on to our next presenter.
Hi, Manan, would you like to go first?
Yeah, sure. Okay. So, Mike, thanks for a very nice presentation. Okay. This is my second interaction listening to you. Last time I had that fascinating CSF flow thing observed, okay, that we didn't see today, but then I was... First of all, let me begin by thanking the Long COVID Foundation and Valentina to bring all of us together, you know, because these type of efforts are the ones, okay, which are needed, and we should always acknowledge the heavy contribution from the Long COVID Foundation that actually brings us together. My question to you, Mike, is that nowadays, okay, you must be hearing a trend of vagus nerve stimulation, okay, in long COVID. My question is that, as you described, the cytokines, they irritate the vagal nerve ending, the vagus goes, and it relays in those centers that you mentioned. My question is that if we do a vagal stimulation, okay, and it actually would transmit signals to the brain, how would it be helpful in any COVID, I mean long COVID or the symptom? At the end of the day, it's a stimulation, be it cytokine or electrical stimulation. So I want your thoughts on this, please.
Sure. Yeah. Thank you very much. And it's nice to see you again for sure. So yes, vagus nerve stimulation is something that people have talked about. I think it's worth pursuing and worth doing. We're trying to get a study going of that to see. Now, I think it's important to note with vagus nerve stimulation that if there's, for example, if there's viral reservoirs, for example, it won't address that problem, right? It doesn't necessarily get to the root cause in that circumstance, but I think it can be helpful. If there was a clearance, for example, the vagus nerve is really interesting because it goes in two directions. So from the body to the brain, the afferent or sensory direction detects inflammation and sends a signal to the body, from the body to the brain that says you're sick, feel sick, go to bed, etc. In the other direction, there's what's called an anti-inflammatory reflex. So in the efferent or motor direction of the vagus nerve, there's a synapse in the spleen that ends up producing anti-inflammatory cytokines. There's different types of cytokines, right? So the thinking is that with a vagus nerve stimulating device, you could theoretically be sort of regulating the afferent direction, a little bit like a pacemaker. I don't want to stretch the analogy, but a little bit like a pacemaker in the heart, where it's sort of, trying to regulate what might be a spastic or ongoing signaling a little bit. And then also at the same time, hopefully exercising that motor or efferent direction and the anti-inflammatory reflex. So there's pretty good evidence, for example, in sepsis, where that's a really extreme example of inflammation, right? A genuine cytokine storm that can be deadly, probably much more than what people with long COVID are facing. But even with sepsis, it increases survival times, probably because it's regulating both directions of the vagus nerve. So yeah, I mean, I think it's worth noting, and most of the I don't have any relationship with any of these companies. I talk to them, but I don't have any investments or anything. Most of the vagus nerve stimulating devices that I think I are most comfortable with tend to focus on the ear. There's what's called the auricular branch of the vagus nerve. It's the place of the vagus nerve that comes closest to the skin of the ear, and they stimulate right up here in the cymboconcha or the tragus of the ear. They tend to use the right side. They tend to use the left side of the vagus nerve so that it doesn't affect the heart. But yeah, vagus nerve stimulation is something that I think is really worth investigating, especially the ones that stimulate near the ear.
One more thing, okay, a follow-up question to your presenting slides, okay, one of which showed that blood-brain barrier thing. My question is that with the COVID infection, and when I say COVID infection, I mean active COVID infection, won't the blood-brain barrier be already breached by the SARS-CoV-2, okay, as it passes through cerebral circulation, slowly interacts with the endothelium? So if it's already breached it, those cytokines that you were mentioning, okay, they are in the blood. They are just like an easy track to go to the brain. So what are your thoughts that initial SARS-CoV-2 infection can affect the blood-brain barrier very initially in the stage of the infection, and then the cytokines have got a free pass to cross across?
Yeah, I mean, the SARS-CoV-2 virus is capable of neurotropism. We know that. Whether or not that's what's causing most deaths, I think, is probably maybe not so clear or less likely, but it clearly is capable. And the circumventricular organs, the choroid plexus, for example, are unfortunately a place where the virus can infect and have an effect. And the choroid plexus they're circumventricular organs that are the place where cerebrospinal fluid is produced. And this is an example where the virus, to the extent that it can get into blood, can end up interacting with the brain and penetrating into brain. And yes, you'd have two problems there. One is that the blood-brain barrier would be disrupted or enlarged in that circumstances. And then the actual just cells right there would be producing cytokines as well, right, locally, which would have a profound effect on the brain as well. So yeah, it's sort of bad news if that happens, I think. And blood-brain barrier is something that I think is an area of research that's really important to understand long-term consequences. The brain is very demanding of oxygen. It uses a lot of vasculature. Okay.
So, very quick, last question, we know that responses like fever, actually we evolved it evolutionarily. They help us, by providing us rest. My question is that if a cytokine transmits impulses via vagus to the brain and it alters our behavior, that it wants us to rest more, isn't it a defensive thing? I mean, removing it, do you think is a good choice, or you think that this evolutionary thing should be kept upon for some time? Because it's a thing that we developed as a defensive mechanism.
Yeah, I mean, I agree with that. I think it's a tough thing for people to understand, is that something that is subjectively bad, like pain, is there for a reason, right? It's there as a defense. So is sickness. The very feeling of being sick is evolutionarily preserved for, hundreds of millions of years, because it is a signal. It's the behavioral aspect of the signal. Go to bed, you're sick. Fever is a good example where it's part of the innate immune response. If a pathogen has evolved to replicate in a host, it's probably evolved to replicate it at 98.6 Fahrenheit or 37 Celsius. So raising the temperature makes it a little bit harder. Cytokine to brain signaling through vagus does mediate fever as well. So yeah, I mean, I don't think you want, this is sort of my problem with immunosuppression in general. At some point, you have to immunosuppress. If someone's going to die of a cytokine storm, then you have to save their life. But I'm not so sure that that's the right call with the sort of chronic lingering symptoms, because it will make people feel better, which is the tantalizing thing, because feeling bad is part of the immune response, but it may not address the actual root causes. And those ought to be addressed better if we're going to actually get people better, instead of sort of temporarily making them feel a little bit better.
Thanks, Mike, for your great presentation. Really enjoyed it. Dr. Shankara?
Thank you, Dr. Baig. Dr. Elzakker, I got a few questions. We've seen that Omicron has a propensity for neurotoxicity. I've seen a lot of patients presenting with neural symptoms, burning of hands and feet. There seems to be a spinal nerve root involvement in some, where they developed a sciatic kind of pain, those kind of presenting symptoms. And it ranges to a sudden onset of from patients becoming very depressed to mild psychosis and psychiatric issues. And we've established that we have a cytokine-like or a hyperimmune response after about a week of initial viral infection. Now, the cytokine release is seen to be the trigger for these peripheral neuropathic kind of symptoms. And these kind of patients respond well to suppressing those cytokines, and it tends to suppress the pain, being the root of the problem itself. But with vagal nerve stimulation, causing the sick feeling, which is usually a physiologic process in trying to help you recover, is it your contention that this mechanism is because of prolonged inflammation somewhere else in the body, that the continual vagal nerve stimulation is causing the prolonged feeling of being unwell?
Right. So that's a good series of questions there. So obviously an ongoing inflammatory signaling will cause the sensation of being sick. That is sort of a straightforward possibility. But because of the phenomenon of glial priming that I mentioned before, that can be quite oversensitized. So glial cells in the central nervous system are relatively long-lived cells. So they can live for something like 8 years, some different kinds. They're replaced by their own replication. They're not replaced by circulating stem cells. And so there's probably some phenotype preservation across time. So in other words, if the glia become primed from an initial inflammatory insult, they're probably going to stay primed for a long time. And so I think that one of the things that can happen is if someone has a primed glia, that what normally most people could handle in terms of gut dysbiosis or ongoing problems probably will be a little bit sensitive in those folks with a primed system. So I think that is an example of where the ongoing signaling from body to brain through vagus doesn't really have to be all that profound, I would suspect. So that's one of the things that I think. And in terms of the neuropathy, it is true that there's an inflammatory component to that. Neuropathy means, for people listening, there's nerves way out near the skin that are important for pain. They're called C fibers. They're unmyelinated nerves. And, there's evidence from Anne-Louise Oaklander's lab, for example, and Peter Novak, two of our colleagues here at Harvard Med, that the C fibers way out at the skin, the intersection of the dermis and epidermis, become damaged and start to retract. Almost certainly inflammation is part of it. There may be other mechanisms that we're looking into as well that I think could be interesting. That may be part of why there's part of the diffuse pain that people can feel, sort of soreness and things like that. Unfortunately, that can happen way out at the skin or anywhere along the pathway all the way up. The increased problems can be caused. So the pain pathway goes through the dorsal horn, the dorsal horn of the spinal column. There's some evidence for inflammatory signaling there as well. So if there's ongoing inflammation, glial activation, maybe viral reservoirs from this or other viruses that are, 90% of humans have herpes viruses in our body. They like nerves. If there's herpes there, that could be causing inflammation. So, yeah, I think that the pain and neuropathy can be caused way out at the level of the skin. It can be caused at the level of the spinal cord, and can be caused in like an amplifier at the level of the dorsal reticular nucleus or anywhere along the way, or the periaqueductal gray, which is another area of the brainstem that's involved in signaling. So if the glia are acting like amplifiers, turning on nerves, turning on neurons, then that can enhance signaling to cause hyperalgesia. Something that wouldn't normally hurt can end up feeling painful. I hope that's a helpful answer to your question.
Secondly, Carlos mentioned the production of certain neurotoxins, conotoxins, PLAs. These might have more direct effect on acetylcholine receptors, the nicotinic and the muscarinic. Will there be an interplay in neurologic illness with long COVID between the exposure to these directly acting toxins and, of course, the global inflammation they can cause and the gut dysbiosis we see? And so we'll have a mix of different mechanisms causing the neuropathies that we see, both central and peripheral.
So if there are some sort of metabolite or mediators that affect acetylcholine signaling, that would, of course, have profound effects on vagus nerve. When acetylcholine was first discovered by a German physiologist, 100 plus years ago, he first named it Vagus because it was the stuff of the vagus nerve. So acetylcholine is a really profound neurotransmitter for vagus nerve signaling, and is especially in its sort of parasympathetic output, control over heart rate and things like that. So if there's something that's directly affecting acetylcholine, some toxin or metabolite, then that would really, I think, profoundly affect the function of this system in a way that you'd have to address in order to get people better.
Thank you. Thank you for a great presentation.
Thanks for the question, guys.
Thank you very much for everyone here. And again, we'll ask you all to hold on, but we're going straight away into our next speaker. And I'll take you gentlemen kindly out of the screen. to Dr. Leo Galland from the USA. So, Leo, I have the pleasure of being one of your panelists, so I look forward to your presentation, and I'm sure I'll have a number of questions for you.